This is a production of Cornell University.
Okay, so today I just want to talk to you guys a little bit about my work on trying to model a couple of different chromatin states across species directly from DNA sequence. And I'm using a flavor of deep learning model known as a recurrent convolutional neural network. But before I get too deeply into that, I wanted to contextualize why we might want to predict chromatin state in the first place. So we know that accessible chromatin regulates gene expression, and most of the genome, most of the time in vivo, is tightly wrapped around histones and heterochromatin. But transcription factors bind to this accessible chromatin, this open chromatin, go on to regulate gene expression. We also know that not all these regulatory regions are close to the genes that they're targeting. They can be up to, for example, in maize, up to 100 kilobases away. So it's important not just to localize to accessible regions close to genes, but also figure out where they are in distal portions of the genome. And so to put this all together, we can then hypothesize that variation within these accessible regions will lead to variation in transcription factor binding, which will lead to variation in gene expression and then go on to influence all the phenotypes that we care about. So as a more concrete example of this, there was a study in maize across 40 different traits in 2 populations. What they did was they took the partition of the phenotypic variants that's related to genetics, and they partitioned that further down to SNPs that were associated in different annotation classes. What we can see here is that across both populations, about half of the variation is attributable to SNPs with encoding regions. And we expect this. We know protein sequence is important. But what's also striking is that variation within accessible regions is almost as important as variation in encoding. So this suggests to me that it's also very important to identify where these accessible regions are if you want to include them in prediction models. Another important chromatin state is DNA methylation. And so a recent paper in grasses showed that if you look at the chromatin state across different tissues, you can see that they're all unmethylated, the accessible regions. And so what we also know about DNA methylation state across developmental time is it's very stable. And so the suggestion is that if we can predict DNA methylation in a specific tissue, then that could potentially capture the global, potentially accessible space across all tissues. So predicting chromatin accessibility and DNA methylation directly from sequence could have a number of different benefits. The first one is that a sequence-based model could be queried to highlight functional sites and rank alleles. It could be similar to a BERT score and really just serve to narrow down to those top X variants based on effect size. The second important point is that if we get good cross-species models, then we'd be able to predict regulatory regions in species that we haven't done these assays in, and we only have genome assemblies. And this could save time and money by avoiding these relatively expensive chromatin assays. And so we know that these accessible regions are expected to be enriched in transcription factor binding sites. And so we might use that as a signal to separate accessible from inaccessible. But unfortunately, it's a very noisy signal because we observe that there's a lot of spurious matches to transcription factor binding sites in inaccessible regions that aren't functional. And so my hypothesis was that maybe it's really the order and relative orientation of these motifs that serve as a stronger signal to help differentiate accessible from inaccessible. And what we also know is that these transcription factor binding domains are conserved across long evolutionary time periods. And so if we find motifs in one species, we expect them to be observable also in other species as well for transcription factor binding. And so convolutional neural networks have already been shown to be able to predict chromatin state from sequence within species, at least in humans. So we see at the bottom is DeepSeq, which is a pretty standard convolutional neural network, which is one of the first applied to try and predict this problem. And it gets a range of what are known as AUPRs. You can just think of as a metric for classification performance from 0 to 0.8. And then the year after that, DanQ was released, which is a recurrent convolutional neural network, which shows improvement across almost all the test cases, the chromatin states in the tissues that they tested. And so my question was, can we then use DanQ to predict chromatin accessibility across species? So the data that I used to train the model was ATAC-seq data, which basically causes read pileups underneath accessible regions of the genome. And then you can take what's known as a peak color to then binarize this output and get binary accessible sequence and accessible sequence regions. And then I take what's known as UMRs, which you do methylation sequencing on the genome, and you get the methylation state for all three contexts of DNA methylation. You say, if it's absent in all three contexts, that's unmethylated. And so then again, I have binary unmethylated and methylated regions of the genome. So in total, I have training data from about 12 different species for ATAC-C, spread nice and evenly across the angiosperms. For the UMR, we have about 10 species, 5 of which are from the original Grasses paper, and 5 of which were newly culled for this work in the dicots. So just a quick overview of how I'm going to model this. I'm taking in 600 base pairs of sequence, and I'm centering it on the UMR or the ATAC-C peak, and if it's shorter than that, I'll extend symmetrically from the midpoint. I'll feed it into DAN-Q, which is that recurrent convolutional neural network, and I'll attempt to predict 0 or 1, inaccessible or accessible, methylated or unmethylated. I'm interested in comparing, can this model, we know it works within species in humans, but can it do as well across species, in species it hasn't observed? And for now, I'm going to shorthand call this model A to Z for Wrightsman to CF. So how does training A to Z look like for a within species model? So let's say you want to do it within a Arabidopsis thaliana. So we're going to take a holdout chromosome as a test set. And we're going to assign these open and closed unmethylated methylated bins on that chromosome to that test set. We'll assign the rest of the training set. Then I'm going to downsample my training set to make sure they're equal across open and closed, as this is known to improve classification training. After that downsampling is done, my training set looks like this in terms of magnitude: 26,000 observations per species with accessibility and about 36,000 for methylation. Then when you evaluate the model in the test set, it looks something like this. For accessibility, we get an AUPR of about 0.6 in Arabidopsis. And for methylation, a little bit slightly better at 0.65. So for cross species, we're going to choose a test species as our test set. And we're going to take all of those within the test species as our test set. And then assign the rest of the species into the training set. We're going to downsample our training set. And we're going to train the model and evaluate it. And now we get a comparison. How well does my within species Arabidopsis model do to the across species model that hasn't seen Arabidopsis sequence at all? And so when we look across both of these features, we see within accessibility, it's quite competitive. It does almost equally as well within species as it does across species. Less so the case for methylation, but still very close. All right, so here are the results across both features and all my species in my data set. So what's very encouraging is that at least in accessibility, we see a very competitive comparison between the across species and within species models. It's either equal to or greater than the ability of the within species model, particularly in the grasses. If we look at methylation, the case is not as strong, but it's still there across all the species that I've sampled for. Another thing I want to look at is whether or not, or how large the effect size is of a certain transcription factor. And so the way I can do this is by using a method called global importance analysis. The way that works is I'll sample a bunch of random background sequences. In my case, I did 1,000. And I use the base pair frequencies in accessible regions across the genome to sample these. And then what I do is I embed a Jasper motif, which is a database of about 530 plant TFs. And I just ask the question, what's the difference in prediction? I do this for all 1,000, and I take the pairwise differences and I average that. And then I embed the motif at all possible positions. And then I take the maximum of that. And that's my maximum global importance score for a given TF in a given species, in a given chromatin state, accessibility or methylation. And I can rank these TFs by that value. And I get a table that looks something like on the right. So the first thing to notice is that on accessibility between Arabidopsis and maize, we don't see as similar of a pattern. So we do see the TCP motifs in Arabidopsis are highly ranked for accessibility. We see in maize that it's also TCP is highly ranked in the top 10, but it's also these dock type motifs. For the methylation side of things, it's more consistent. We see basically that these AP2 ERF motifs are highly ranked consistently between both species, as well as these BVR BBC family motifs. So in conclusion, I've shown that these recurrent neural nets can model chromatin accessibility and DNA methylation from sequence and across species. These across-species models are competitive to models trained only within species, at least within the angiosperms. And the TCP family motifs we saw ranked highly in global importance to the across-species accessibility models. But for the methylation models, it was AP2 and ARF. And I'm also happy to report that there is a preprint available that shows what I've discussed here and also more on bioRxiv. I've uploaded the data of the models to Zenodo, which are linked to the preprint. And there's code on GitHub, which includes instructions for making your own predictions in your favorite species if you'd like. And finally, I'd like to acknowledge my committee at Recurrent for their guidance, my co-authors for all the work they helped in making this paper happen, particularly Pete, who called those extra dipot unmethylated regions for me, and as well as the Buckle Lab for all their feedback, and their funding sources and high performance computing projects. And with that, I'd be happy to take any questions. So Salve asks, you got better AUP on Rhodopsis and rice compared with maize and other species? Does this mean the genome complexity is playing a role? Yeah, that's a great question. I didn't show it here, but I do notice a trend. So as genome size increases, my performance decreases. And what this boils down to is that the model, I also didn't show this, it makes more false positive mistakes than it does. So what I mean by false positive is the model calls it open, but the data is labeled as closed. And so in these larger genomes, that effect is a lot more pronounced. And so I get less of an accuracy in those species. There's a number of reasons why I think this is the case. First, the ATAC-seq in the angiosperms is called very conservatively. So there's a potential that the model is calling things that are actually open in vivo, but just weren't able to be captured in the conserved peak calling sort of constraints. Yeah, and there's also a number of other reasons, but I hope that answered your question.
Do you have a sense of how additive these motif effects tend to be?
They should be fairly additive. Basically, it's been shown. So what's more important, though, actually, is these motifs have been shown to be additive to an extent that they can sort of interact with each other at the regulatory site. But what's more important is that they're shown to be redundant. So if you start deleting these motifs, it doesn't necessarily mean that expression will be impacted. And so that's also a key point to keep in mind.
This has been a production of Cornell University, on the web at cornell.edu.